Groundbreaking results from a Phase 2 clinical trial presented at SITC 2025 are shaking up the world of cancer treatment. Marengo Therapeutics has unveiled impressive data on their lead program, Invikafusp Alfa, showcasing its potential to combat cancers that have become resistant to existing PD-1 therapies. This is a huge leap forward in the fight against cancer. Invikafusp Alfa, a cutting-edge treatment, demonstrated remarkable activity as a single agent across a wide range of cancers in patients whose tumors had stopped responding to PD-1 inhibitors. The study included patients with advanced solid tumors, and the results are truly encouraging. Here's what the data revealed. Impressive response rates, an overall response rate, ORR, of 20% and a disease control rate, DCR, of 80% were observed in tumors with high TMB, tumor mutational burden, across 7 major solid tumor types. These included colorectal, lung, breast, gastric, gastroesophageal junction, GEJ, head and neck, and bladder cancers. This means that a significant number of patients saw their tumors shrink or stabilize, even after previous treatments failed. Consistent activity across the board, the treatment showed consistent activity across all patient subgroups, regardless of their prior PD-1 treatment history. Even more impressive, 59% of patients experienced a reduction in the size of their target tumors. This is a testament to the drug's broad effectiveness. But here's where it gets controversial. Marengo Therapeutics CEO, Junsu, MD, MBA, highlighted that the data supports their vision of developing a next-generation immuno-oncology (IO) backbone therapy. The company believes Invikafusp Alfa can overcome PD-1 resistance by selectively activating and reprogramming V6 T-cells within the tumor microenvironment. Let's break down the key findings. Pan-tumor activity. Invikafusp Alfa showed activity in PD-1 resistant TMB-H patients across 7 different cancer types. This includes gastrointestinal, GI, tumors, 28% ORR, 78% DCR, colorectal cancer, CRC, 33% ORR, 67% DCR, and non-small cell lung cancer, NSCLC, 20% ORR, 80% DCR. Tumor shrinkage. A significant number of patients, 59%, experienced a reduction in their target tumors. Activity in various patient groups. The treatment was effective in both patients who had and hadn't previously received immune checkpoint blockade, ICB, and across both primary and secondary PD-1 resistant tumors. MSI status. Activity was observed in both microsatellite stable, MSS, and microsatellite instability, MSI-H tumors, demonstrating clinical benefit regardless of MSI status. Safety is always a critical factor. The safety profile of Invikafusp Alfa was consistent with its mechanism of action, with treatment-related adverse events, AES, being transient and manageable with supportive care. And this is the part most people miss. Mechanistic activity was validated through paired biopsies and blood samples. The results showed selective V6 T-cell expansion, both in the periphery and within the tumor. In vivo TIL reprogramming with increased TCR signaling, cytotoxicity, and chemokine expression, indicating enhanced immune activation. Downregulation of immune-suppressive factors and upregulation of activation and cytotoxic markers among patients who experienced tumor shrinkage. Invikafusp Alfa is designed to selectively activate and expand V6, Vβ10 T-cell subsets, which are the most prevalent tumor-infiltrating lymphocytes, TILs. This approach aims to restore and amplify anti-tumor immunity in patients who have become resistant to ICB. Marengo Therapeutics is taking a strategic approach to develop Invikafusp Alfa, focusing on biomarker-enriched development and exploring combination therapies. They are also working closely with regulatory agencies, including receiving US FDA Fast Track designation for metastatic colorectal cancer, mCRC. What are your thoughts on these findings? Do you think this approach could revolutionize cancer treatment? Share your opinions in the comments below.